Alright, so I chose these two papers because they are about the circadian clock and health in all its incarnations. The paper titled Circadian Rhythms, Time-Restricted Feeding and Healthy Aging is about what happens when the clock breaks down in humans or in animal models like mice. It's a fairly recent review about these things, so we are going to start with this one. And then the next review, it's a big, as you can see, many pages. We don't need to see everything because this is the last lecture. But you can see the authors, Francis Levy and Ueli Schibler. So Levy is a French person and Schibler is somebody from Switzerland. And Francis Levy is a physician who used to work in France. And he's the first person who has proposed to take into account the internal time of the patient to treat cancer. So this is not about what happens when the clock breaks down, but this is about how the clock can help in treating diseases, especially like cancer. Okay, so we are going to see that later. So let's start with the circadian rhythms, time-restricted feeding and healthy aging. The first page you don't need actually to see. We will not see it together because it's again explaining about the molecular clockwork and everything. You can read it if you want by yourself, but we have already seen most of what happens. Okay? And we are going to skip down to the point number the point so you can, with your pen, you can underline 3.2. So this is on page 3, the point 3.2, 3.2.1, 3.2.2, 3.3 we have already seen, and then 4 and 5. This is what we are going to see about this paper. Okay? All right. And we are going to read as usual because I think it's a good thing to do that. And we are going to start. Maybe I will start. Okay, so this is the point 3.2, the endocrine system, which is something that I have not really gone into in this lecture. So, insulin and glucagon. The pancreas regulates blood glucose by producing and secreting insulin and glucagon to control release of glucose from the liver when blood glucose levels are too high or too low, respectively. Insulin is produced from beta islet cells in the pancreas with weak production around 5 hours in the afternoon and a nadir around 4 hours in the morning in humans. Temporal control of hormonal production and release is controlled by both feeding-fasting pattern and circadian rhythms. Eating patterns greatly alter nutrient levels in the blood and can therefore act as an acute overriding signal. The circadian system modulates both insulin and glucagon by controlling production and secretion at the cellular level as well as SCN signaling the autonomic nervous system. In vitro, beta islet cells exhibit robust rhythms of both BMAL1 and PER1. BMAL1 knockout in the pancreas has disrupted glucose homeostasis and insulin release despite displaying normal activity and feeding fasting rhythms. This demonstrates how the molecular clock at the cellular and tissue levels can have significant level on physiology. Insulin sensitivity is also influenced by both nutrient state and the clock through SIRT. Research in both animals and humans has shown that caloric restriction increases level of SIRT. Moreover, mice on an ad-lib high-fat diet dismarket. So we can actually skip this SIRT one. There is a mention of SIRT earlier in the paper. This is about the 0.31 metabolite. So we'll not go into detail with this one, but let's just say that SIRT is a protein that controls gene expression. It's a transcription factor that reacts to the energetic level of the body. And when the energy is high or low, then SIRT reacts and will regulate gene expression. And it's one way by which the energy level can actually regulate the clock. Okay? Because SIRT will regulate clock and BMAL and PER2 transcription depending on how the energy of the body is. So let's just say that for the point 3.2, this is the fact that all these enzymes like insulin and glucagon, yes, of course, they react to the food intake, but they're also controlled by the clock. So there are two influences of this system. Okay? And if you have a question, please ask. Otherwise, we are going to see the point 3.2.2, which is the cortisol. The cortisol is one of the first hormones to be demonstrated to have a clear circadian pattern of secretion. Okay, so first of all, do you have any question about insulin and glucagon?
Well,
What is nadir?
Nadir is the lowest point, yes.
What is active? On the second paragraph, active high fat fire. Moreover, mice on the second paragraph. Moreover, mice on the active high
Moreover, ad-lib, what is ad-lib? What is your question?
Yes.
What is ad-lib?
Is what is active?
Ad-lib means yes. Normally, the full name, Kagaku Igo no Hanashi. The full name is ad libitum. Ad libitum means you can have access food whenever you want. It's not limited. But some scientists, they just remove ibitum and they just say ad lib. As much as you want, not limited, at any time.
it's
Okay.
ad, not ab.
Eh?
This
They write
shows,
ab lib?
Adlib.
yeah.
Really?
Yeah, that's the saying, adlib.
This
They write
paper
ab lib?
say-
Yeah.
Really? I didn't even see.
Yeah.
Ab lib is bad. It doesn't,
It's a
it
mistake.
doesn't exist. This is ad lib. Okay? This,
When people use it, we normally say adlib. We don't say adlib.
really?
Yeah, we say adlib.
Ab lib.
Yeah.
But this is wrong. This is ad libitum.
Oh, but we cannibalized it.
Really? Really?
Yeah.
And this is bad. This is bad. So the correct ab lib is apparently okay, but the correct one is ad libitum. Ab lib. So good. Actually never seen ab lib.
It's used a lot, like in.
It's the first time I see.
Really?
Yeah.
We say so I normally know it in like the comedy context where you go off ad-lib is when you kind of just make stuff up on the spot.
Really?
Yeah.
That's weird. That's weird. All right. Okay. So it also exists apparently. Happy. All right.
It's curious that BMAL1 knockout in the pancreas had disrupted glucose homeostasis and insulin release despite displaying normal activity and feeding fasting rhythms. Feeding fasting rhythms is overriding signal, and pancreas somehow controls the timing of releasing. Why this pancreas disrupted rhythm?
Yeah, so that's a very good comment. So far, what we have seen in this lecture is knockout animals in the whole body. So you remove BMAL1 in the whole body, and if you check locomotor activity of the mouse, or if you remove CRY1 and CRY2, then you completely lose rhythm. But of course, sometimes you get problems in metabolism and things like this, but it's difficult to know the mechanism because all the body is affected. So why? So then, this is very interesting to do the same work in which you actually do knockout only of specific tissue. And this is great because even though the SCN of the animal is intact, genetically intact, so it has a normal light-dark cycle rhythm, and it has a normal rhythm in constant darkness. It will eat at the right time, it will fast at the right time, but still you will see disturbance in specific tissue. Like in this case, for example, even though the animals have a normal activity and feeding fasting rhythms, the glucose homeostasis of this animal is disrupted. And what people say, and I think this is quite important, people will say that the lack of the biological clock in the pancreas will affect glucose homeostasis. Do you think that's correct? You remove BMAL1 from the liver, from the pancreas or from the liver or whatever, and then you say, okay, so when we remove BMAL1 from the pancreas, this means that without the clock, glucose homeostasis is perturbed. Is it correct to say that? Think about it. Yeah, this is what many scientists say. This is what many scientists say. This is what many scientists say, but actually the more correct is that BMAL1 is required for glucose homeostasis. You don't know that these changes in glucose homeostasis is because you lack the clock. Maybe it's because you lack BMAL1. Because mice who are CRY1, CRY2 double knockout mice, they have different problem of the pancreas. The clock is not there. They are also arrhythmic, but it's different. So this really means that BMAL1 is also involved in maybe the circadian control of glucose homeostasis. But it is very dangerous to say that removal of the clock cause problems, because you don't know if it's really removal of the clock. It's removal of BMAL1, yes, you cannot say anything against this. So this is actually one of the comments we got in our recent paper that we submitted in the laboratory. Removal of a clock gene doesn't mean you remove the clock. It just means you remove the clock gene. Okay? And the function of that clock gene is maybe to control insulin sensitivity or glucose homeostasis and things like this. So even despite the fact that the animal has rhythms of food intake, still, if it lacks BMAL1 in the pancreas, there are still some problems there. Okay. Let's go for the cortisol. Next, please.
Cortisol is a steroid hormone in the glucocorticoid family that is involved in metabolism and stress response. Production and secretion of cortisol are rhythmic and regulated by the hypothalamic pituitary axis, HPA, and the autonomic nervous system, ANS. In the HPA axis, corticotropin-releasing hormone, CRH, and arginine vasopressin, AVP, are produced in the paraventricular nucleus of the hypothalamus. CRH is released from the median eminence and travels to the anterior pituitary through the hypophyseal portal system. Hypothalamic AVP neurons project axons directly to the posterior pituitary and release AVP into the circulation. In the anterior pituitary, both CRH and AVP stimulate the release of adrenocorticotropic hormone, ACTH, into the blood. ADP in the adrenal medulla also increases adrenal gland production of ACTH. ACTH acts on the adrenal cortex to stimulate cortisol release. This system is negatively regulated by cortisol in the hypothalamus and the pituitary. The PVN also sends ANS projections to the intermediolateral column of the spinal cord, which connects to the splanchnic nerve to signal the adrenal gland. Both HPA and ANS regulatory pathways for cortisol are regulated by the SCN at the level of the PVN and upstream in the dorsal medial hypothalamus and subparaventricular zone. Cells of the adrenal gland also have cellular clocks that temporarily influence production and release of cortisol.
Okay, so this is a difficult paragraph. So the first part of the paragraph just explains this HPA axis, which is a very, very famous axis for the control of hormone synthesis. So the first step happens in the hypothalamus, and then this controls the release of ACTH from the pituitary gland. Okay. Then the pituitary gland, the hormones from the pituitary gland, they influence the adrenal gland to produce cortisol. So the whole pathway here is also controlled by the SCN. Okay. So that, what do you think happens? So the final step is cortisol. Okay. They explain how cortisol is controlled by the hypothalamus and the pituitary gland, but in addition, the SCN controls this axis, which results in what? What do you think this results?
Rhythmic cortisol expression?
Yes, very strongly rhythmic cortisol expression. Not only cortisol has a very strong circadian rhythm, but it is also a hormone that dramatically is induced by stress. So let's say you take a mouse and simply you talk loudly, or you take a mouse and after only a few minutes the cortisol will be very, very high. So this is one problem, because if you actually want to quantify cortisol in mice, it's very difficult to do it properly, because the stress of the mouse because of your sample will actually make a lot of problem. Okay. But if you do that right, then you will see that cortisol has a very strong rhythm of expression. And I think there is...
Does it happen every time, or can mice get acclimatized?
What do you mean? Acclimatized to stress?
No, to like human presence, because people have pet mice.
Yeah, so of course there is habituation, okay, and some mice will habituate differently than others, but there is habituation. But this is not something you want for your mice in the laboratory. So this is the problem, is that you want to, especially for circadian thing, you want to put them in a dark cage, and then, so if you want to sample at different time, it's very, very difficult. So, but it's possible to do. You know, if you try to limit the amount of stress you give the animal, and then, so it can be a problem. It can be a problem. But the bottom line is that cortisol has a very strong rhythm of expression, because cortisol actually is part of the awakeness of the animal. Okay. Then let's continue with the same paragraph, and that will be Nguyen-san, please.
Glucocorticoids are released with both an ultradian 1 to 2 hours and circadian rhythmicity. Peak levels of glucocorticoids synchronize with the beginning of the active phase to aid in arousal, early morning in diurnal animals and early night in nocturnal animals. Likewise, ACTH exhibits a parallel rhythm, and it is also modulated by the SCN. Similar to negative feedback on the HPA axis, glucocorticoids influence circadian rhythm by feeding back on the clock. Glucocorticoids bind to a glucocorticoid response element, or GRE, in the PER1 or 2 and reverse alpha or beta promoter to activate and suppress transcription respectively. Conversely, CRY acts as a transcriptional suppressor of glucocorticoid regulated genes.
Okay, thank you very much. So that's easy enough. So this is what they say, right? So I think this is the first time we see the word ultradian. Ultradian is a rhythm that has a period less than 1 day. So typically, if you have 12 hours rhythm, this is ultradian. If you have 2 hours rhythm or 3 or 4 hours rhythm, this is also called ultradian. It has no indication of any specific duration. As long as it's lower than 24 hours, significantly lower. If it's 22 hours, we can still say it's circadian. But if it's like about 12 or below, this is usually ultradian. Okay, so not only the clock controls the secretion of glucocorticoid synthesis, but glucocorticoid also feedback on the clock to regulate it. So the whole organisms make sure that it is synchronized at the level of the whole organism. Okay, so that's just to set up what we are going to see later. And as you read, so in relation to the cortisol being stress hormone, so that promotes awakeness, then it's not surprising that the peak levels of glucocorticoids are high during the active phase. Okay. But that's different in human and mice. In mice, glucocorticoid will be high at night, and in human, they will be high during the day. All right. Okay. So then we can shift down to 5 first. And maybe you can repeat.
5?
5. Consequences of circadian disruption.
In humans, the most obvious consequence of circadian disruption are observed in activity-rest cycles. Changes are also seen in amplitude and phase of temperature and melatonin rhythms, yet some studies show little to no changes. Sleep quality and consolidation are also greatly disrupted. Genetic, behavioral, and lesion-induced circadian disruption or arrhythmicity has been shown to result in a wide range of health consequences, including increased risk for cancer, cardiovascular disease, obesity, immune disorders, infertility, and affective disorders. Genetically arrhythmic mice, BMAL1 knockout, exhibit symptoms of early onset aging, including a decrease in muscle and subcutaneous fat, cataracts, and organ shrinkage. Circadian disruption caused by shift work in humans is associated with cardiovascular disease, metabolic disorders, and cancer. In rodents, transplanting the SCN from a young hamster to an old hamster with weak behavioral rhythms was sufficient to not only reinstate robust behavioral rhythms in the older hamster, but also increase the lifespan by 4 months. Normal lifespan is about 2 years. This indicates that aging of the SCN also dictates aging of behavioral rhythms and physiology.
Okay, thank you very much. So that's very interesting. And to a certain extent, it is linked to the point 4, that the circadian rhythms dampen with age. So if you, for example, check the SCN of young versus old animal, the amplitude of circadian rhythm of the SCN, so you can think about gene expression, or you can think about neuronal activity, they will dampen with age. So the older the animal gets, the more dampen the rhythms are. When say dampen, it's more like amplitude. So if you check, for example, any circadian rhythm in a young animal, you may get something like this. But if you actually check the same rhythm in an old animal, it may become very low amplitude. Okay, and very interestingly, in this experiment from 1995, if you actually take a SCN from a young animal and put into an old animal, this sort of elongates the lifespan of the old animal by 4 months. Presumably, just because only putting a very healthy, high amplitude, with a high amplitude rhythm is enough to regenerate all these cycles of things in the body. Okay? Why do you think this might be? Why do you think this might be? I mean, suddenly you can imagine that because the old animal inherits from a healthy SCN that has high amplitude rhythm and that efficiently controls the body. Okay? Because SCN will be in the brain, even though it's implanted, it will also control all these things that normal LCS do. So can you actually tell me why the animal live longer? What could be the possibility of that? What could be the reason? It's not just because of rhythms. This is what they say here, right? They say in rodents, transplanting the SCN from a young hamster to an old hamster with weak behavioral rhythms was sufficient to not only reinstate robust behavioral rhythms in the older hamster, but also increase lifespan by 4 months. This indicates that aging of the SCN also dictates aging of behavioral rhythms and physiology. Yes, okay. But why would putting a SCN in an old animal increase? If you tell me that it's because the old animal suddenly has better rhythm than it had before, this is not an explanation. That's not an explanation, that's the observation. Okay? You put a SCN, you check that the rhythms of cortisol in this old animal, that the rhythms of gene expression in the liver might be more with a higher amplitude. This is not an explanation. This is exactly what happens. What might be an explanation of why these animals have a longer lifespan.
If the rhythms are more pronounced,
Yeah.
then things like cell cycle will be better.
Yes, that might be one of the reasons. Is that maybe since the rhythms have a high amplitude, then all these cell cycle, synchronization of cell cycle will be, it will be more harmonious. And this may help the animal to cope more with cellular stress and things like this. What else?
They mentioned cardiovascular disease and metabolic disorders. For example, the insulin and glucagon release is more soundly controlled, and if with old SCN, they can't control blood sugar, and they are easy to form blood clotting.
Yes, exactly. So these all things are possible. This is maybe because, as you said, the whole energy metabolism is improved, and it's probably one of the main reasons why there is a very big theory now that actually circadian rhythm is a way to be more efficient and to conserve energy. If you do things that are highly demanding in terms of metabolism, you want to do them only when necessary. So you do them during this time. And when you sleep, everything goes down and you save energy and you repair your cells, you repair your DNA or whatever. But if your rhythms are becoming dampened, then suddenly you are left with cells who always have a higher level, not higher level, but they always have the same amount of stress all the time, the same amount of damage, and this may not be so good. Okay, this is only a possibility. It has not been demonstrated why, but these are the possibilities that the whole control of the metabolism is improved, so you keep energy, you save energy, you are more energy efficient, and this may help for survival as well as the cell cycle. Okay. Then let's continue. So I will continue this one here. In addition to an increased risk for disease, circadian disruption can also lead to cognitive deficits. Rodent models have demonstrated that chronic circadian disruption induces deficits in hippocampal learning and memory, but not in fear conditioning. Circadian disruption caused by keeping mice in a short photoperiod, 20 hours, 10-hour light, 10-hour dark, inhibited cognition measured by cognitive flexibility tests. Circadian disruption induced by keeping mice in a short photoperiod, same, a period too short for a wild-type mouse to untrain, inhibited cognition measured by cognitive flexibility test. Am I reading 2 times the same thing?
Mm-hmm.
Yes, I am reading 2 times the same thing. So the sentence from circadian disruption caused to Calat-Soleos 2011 is repeated twice.
Oh.
I had the impression of living the same moment again.
They've not only done that, they've changed one word.
They changed.
That was deliberate.
It is funny. So anyway, they said the same sentence, almost the same sentence twice. These mice also showed a decrease in dendrite length and neuronal complexity in the prelimbic prefrontal cortex, a region of the brain involved in executive functions and emotions. Observations of airline cabin crews have shown that chronic jet lag is correlated with decreased cognition and temporal atrophy. Say that to the cabin. I think you suffer from temporal atrophy. I beg your pardon. Rodent models of jet lag demonstrated an inhibition of neurogenesis in the hippocampus and long-term cognitive deficits and shortened lifespan. There's a lot of problem in this paper. Memory deficits may be explained by the circadian control of brain-derived neurotrophic factor BDNF expression in the dentate gyrus, but not in the CA1 or CA3 of the hippocampus. It is unclear if this control is direct from the SCN or indirect through corticosteroid suppression. BDNF modulates cell growth and survival in the hippocampus, plays an important role Panda is from the United States. He usually writes well, this guy. So anyway, there is a lot of strange English in this paper. It is unclear if this control is direct from the SCN. Memory formation is dependent on long-term potentiation to alter synaptic connections. Increases, okay, so whatever this is about molecular, this is not so important. This is potential mechanisms, it has not been proven. But why I wanted you to read this paper, because not only lack of the clock or lack of clock gene has effect on metabolism, but also on cognition, okay? Like, one of the most important part of this paragraph is that something that I did not explain this year is short photoperiod. So this is circadian disruption induced by keeping mice in a short photoperiod. So normally, the photoperiod is 24 hours. And you have 12 hours of light, 12 hours of darkness. You can change that. You can put 16 hours of light, 8 hours of darkness, or 8 hours of light, 16 hours of darkness. Okay, this will be like winter and summer. But what you can also do is, instead of giving a cycle of 24 hours, you give a cycle of less than 24 hours. If you give to the animal a cycle of 23 hours, for example, 11 hours of light, followed by 12 hours of darkness, still the animal will be able to untrain. It's a 23-hour cycle. If you give 22-hour cycle, so let's say 10 hours of light, 12 hours of darkness, still the animal will be untrained. But if you give less than that, below 20 hours, the animal will not be able to untrain. So the biological clock will try to untrain to this light-dark cycle that is quicker than itself, but it might not be able to untrain. Okay? And what you can do as well is to give constant light to animals. And depending on the animal, constant light may actually cause complete arrhythmicity of animals, and you have strange rhythm appearing. So that means that you are completely destroying the clock of the animal without affecting genetic makeup. Okay? And when you do that, then you have all sorts of funny things happening with cognition and with motor functions and things like this, showing that the clock is actually very important for brain function, which you see as a disruption of the behavior of the animal, not only in the light, not only in the rhythm of activity and rest, but also in how well the animal performs when you ask the animal to do tasks. Okay? So I think this is fairly important. So this is linked with the in human, it's very difficult because you are never sure of anything because it's only by association. But the big catastrophe usually happen during the night. Like, for example, the 3 Mile Island, for example, in the United States. Have you heard of the 3 Mile Island in the United States?
3 Mile Island?
No? So there are typically things like, for example, a nuclear power plant, suddenly, that has a major accident. when you look at how these things happen, they are usually during the hours of night or the morning. Okay, and there are many other. You can take a look. For example, there is the 3 Mile Island. I think I have. So the 3 Mile Island, it's a little bit similar to what happened in Fukushima. And the result of the investigation is that actually the people in the plant did not react early enough to prevent the problem from happening, because it was during the hours of the morning and the people were less able to react to a difficult situation. And this is usually attributed to the fact that when these things happen, when you are less able, then the likelihood of having a problem increases. It's not a proof, but it seems to be a recurrent observation that when there is a major catastrophe, it's always at a time where the response time is much lower. Okay, and you can see there are some reports. There are some reports about the 3 Mile Island. Colline. The 3 Mile Island accident was caused by a nuclear meltdown that occurred on March 28, 1979 in reactor number 1. So this is one of the, just like Fukushima type stuff. And then I kind of wonder if they actually mention human factor. So critical human factors and user interface engineering problems were revealed in the investigation of the reactor control system. Despite the valve being stuck open, a light on the control panel ostensibly indicated that the valve was closed. And blah 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 blah. I want to find a time where this happens. Because yeah. So the radioactive coolant was pumped from the containment billionaire outside the main until the sump pumps were stopped at 4:30 a.m. So very, very early in the morning, where the people were at the most like 4:15 a.m., 4:11 a.m. So these happened very, very early in the morning. And this is not the only such catastrophe. Like I think the Exxon Valdez as well was something that happened. See, this is another type of thing. following the 1986 annual meeting of the Association of Professional Sleep Societies, a committee of scientists was formed to review recent reports and related information on the role of human sleep and brain clocks, time of day variation in physiology and alertness, in the occurrence of medical and human error catastrophe. This is a report of the committee's finding and recommendation. So, because of the fact that the clock really controls via this cortisol system our alertness, then if you are doing something that requires brain power, then it is very dangerous, because when you are required to respond quickly, you may actually not be biologically programmed for it. During the day it's fine, but at night the likelihood of having major catastrophe increases. Okay? So this is linked to this paragraph here that we just read. Okay? You can also observe that in mice, when you force them, for example, to be under jet lag, or when you disrupt their circadian system by strange light-dark cycles, then you can see all these brain functions also decreasing. Okay? So it's not only a metabolism thing, it's also a brain function problem. All right, then let's go to the point 5.1. So I skip on all the molecular things. They are not the main thing here. Okay, 5.1, please.
There are many correlations between circadian disruption and neurodegenerative diseases, such as Alzheimer's disease and Parkinson's disease. However, the causal relationship is unclear, as circadian rhythms have been shown to play a role in neurogenesis. The dampening of circadian rhythms with age is likely to contribute to neurodegenerative diseases such as Alzheimer's disease. The amyloid hypothesis of AD proposes that excess production and accumulation of amyloid beta peptide begins a cascade effect that damages the synapse and neurites and leads to tangles containing tau proteins. Prior to aggregation, there are daily fluctuations in amyloid beta soluble interstitial fluid, controlled by the sleep-wake cycle. Levels of amyloid beta in the ISF correlates with the amount of future amyloid beta aggregation. After amyloid beta plaques are formed, diurnal rhythms of both the sleep-wake cycle and fluctuations in ISF amyloid beta are diminished. In humans, Alzheimer's patients exhibit a decreased amplitude in activity patterns and a delay in the peak activity and body temperature. There is also a correlation between circadian disruptions in locomotor activity and sundowning, an exacerbation of AD symptoms in the afternoon. This is also associated with a delayed peak of body temperature in the afternoon. Further circadian disruption in AD patients can be seen in the SCN. In humans, SCN neurons expressing vasoactive intestinal peptide, VIP, another important intercellular coupling factor in the SCN, was significantly lower in females with AD compared to healthy age-matched controls. Interestingly, this effect was not seen in males. However, young males had more VIP neurons in the SCN compared to older males, whereas young females had less VIP expression compared to healthy older females. Although it is unclear if AD leads to circadian disruption or vice versa, some behavioural modifications using bright light exposure have been shown to restore some behaviour rhythms in patients with AD and dementia. These studies indicate that low amplitude circadian rhythms contributes to the AD and dementia pathology.
Okay, thank you very much. So this paragraph is mainly on the fact that neurodegenerative disease have a circadian component. But the problem is, because it's in human and there are no really conclusive evidence using animal models, it's just a link. There is an association between neurodegenerative disease and the circadian clock. But which causes what? We do not know. Okay? As they said in this paragraph, they said there was one sentence that really synthesized this one. Although it is unclear if Alzheimer's disease leads to circadian disruption or vice versa. Okay? So disruption of circadian rhythms in Alzheimer's disease may just be one sign of Alzheimer's disease, but it's possible that the circadian disruption causes some symptoms of Alzheimer's disease. We don't know which is the first one to happen. Okay. What is clear is there is a circadian component in Alzheimer's disease. So what would you do? What would you do if you know there is a circadian component in Alzheimer's disease? So for example, you have patients and you see that, for example, their sleep-wake rhythms is disturbed.
See if there's one of the EVAX regions on the APEN genes. Sorry. The genes associated with Alzheimer's. You can see if there's the region for the.
Oh yeah, you could check at the molecular level what is happening if. Well, this is what they say, right? There is apparently some circadian components even in the beta amyloid amount, right? So there is maybe some sort of circadian thing, but what I mean is if you have a disease that, like Alzheimer's, for example, that not only maybe there is something happening at the molecular level, but what I want to say, I think the point is that if you have a disease, like Alzheimer's patient, for example, the fact that they have disrupted rhythm, maybe, it's not the worst of their symptoms, right? It's not the worst of their symptoms. But the thing is that if you were able to improve the circadian organization of their sleep, for example, that may make their symptom lessen. So you could act on their circadian rhythm to try to improve their sleep, which would improve their symptoms. This is one thing that you can do. Okay. But at the moment, all these studies in humans, they only suggest that there is a role of the circadian clock on these diseases, but we really have no idea. Okay, so that's the last point of this paper, I think. Okay. Like there is a disease. Do you know DM? Myotonic dystrophy. It's similar. Myotonic dystrophy, I should write it because there are two types of myotonic dystrophy. The main one in Japan is DM2. Myotonic dystrophy. So it's a muscular disease, and the problem is that the people who suffer from myotonic dystrophy and mice who are used as model for myotonic dystrophy, they also have sleep problems. They have sleep problems. The problem is that we have no idea why they have sleep problems. They have a genetic disease which makes their muscle really disappear, but they have a problem in sleep. And in many diseases like this, degenerative disease also usually, this is accompanied by a degeneration of circadian rhythms like sleep. And maybe this is something that it's only a possibility. There is no proof in any of these things. But the people are kind of saying that maybe if you were able to help these people to have a better circadian clock, they would have better sleep, and again their symptoms maybe a little bit decreased. But again, this is not something proven or anything, but it's one possibility. Anyway, so from there, we are going to go to the next paper and something that is more helpful for patients, I think. It's how the clock can be taken into account for the treatment of disease. And we are going to skip the beginning because the beginning is again about the molecular clock, about PERP, CRY, BMAL1. You know all these things already. And we are going to go directly to page 601. The point, chronopharmacology, dosing time dependencies in drug effects. And please start.
The widespread awareness of the importance of the time of day on pharmacology can readily be illustrated by a literature search. As of May 2006, entering pharmacology and circadian into PubMed resulted in more than 14,000 references, including approximately 1,200 review articles. The influence of the time of day on drug efficacy and toxicity is perhaps not surprising, considering that most mammalian physiology is affected by the circadian clock. Currently, most approaches, considering the relevance of dosing time for drug effects, do recommend a standard best time for drug administration in populations with well-synchronized circadian physiology. Recently acquired knowledge on the circadian timing system and the availability of new experimental and computational models and technologies now allow for the identification of the key clock and clock-controlled components that influence the dynamics of drug effects. In turn, the variability factors that impinge on those biological timers and their signaling pathways can now be studied more precisely.
Okay. So that's very simple. Let's continue to read. Nguyen-San.
The level toxicity of a fixed dose of a drug has long been known to vary as a function of dosing time in laboratory rodents, irrespective of drug class. For instance, the lethal toxicity of mice exposed to halothane fluctuates between 5% and 76%, depending on when the animals were exposed to the drug. 2 to 10-fold changes further characterize the tolerability of mice or rats as a function of dosing time for more than 25 anti-cancer drugs, including anti-metabolites, 5-fluorouracil, 5-FU, and L-adenosines, DNA interconnectors, doxorubicin and peptidoglycin, alkylators, cyclophosphamide, and oxaliplatin, mitotic inhibitors, vinorelbine, and doxorubicin, interferon alpha, COX-2 inhibitor, celecoxib, or angiogenesis inhibitor, TNP-470. These reproducible 24-hour variations in drug toxicities have been documented in mice or rats kept in a regular alternation of 12 hours of light and 12 hours of darkness. A photoperiodic regimen that ensures proper inter-individual synchronization of circadian physiology, provided that noise, light at night, or external temperature cycles are avoided. Similar toxicity rhythms were found for doxorubicin in rodents, switched to constant darkness or to constant light, thus ruling out a major masking effect of light or darkness.
Thank you very much. I think that's enough. So that is very interesting. So for many drugs, it was found that depending on the time of administration, the outcome of the treatment and the toxicity can be completely different. And something that is important is that a mosquito just died. What is very important is that not only these daily differences in toxicity of drug were observed in light-dark cycle, but also in constant darkness, which shows that this is not because of light or whatever. Okay. So, give me your hypothesis. Why this happens? For example, let's focus on the first drug that Nguyen-San read, this 5-fluorouracil. What is 5-fluorouracil? This is one of the main cancer chemotherapy.
Replace uracil in RNA.
Yeah, it's uracil in our DNA? RNA, sorry, yeah. Yeah, so it replaces uracil in our RNA, but it is also metabolized. It is also metabolized and is also added to DNA, actually, okay? So it's a nucleotide analog that actually interrupts DNA synthesis, okay? So why do you think, well, the same thing can be said for all the other drugs, like oxaliplatin and things like this. So why do you think the toxicity of drug changes? I mean, if you inject 5 milligram per kilogram of body weight to an animal of 5-fluorouracil at 10 o'clock in the morning or 10 o'clock at night, it's the same amount of drug. The drug doesn't change, but just the time of administration will make a lot of difference in the toxicity. Why is it? Why is it? Why do you think it is?
Concentration of the enzyme that stimulate the metabolic reactions changing during the day, so it will have different effects.
Yes, that's right. Can you maybe guess what kind of enzyme? I mean, not what kind of enzymes, but what tissue will be most important for that? For the pharmacodynamics, what happens to the drug once you inject to a person? What is the main tissue? Huh? Yeah, the liver is very important because the liver is a tissue, it's an organ that will detoxify many of the xenobiotics, molecules from outside. So if the metabolism of the liver has a circadian rhythm, then there is no surprise that when the liver sees 5-FU or oxaliplatin or whatever drug, the liver may be very quick to get rid of the drug at one time, but will be very slow at any other time. So this explains that the toxicity of drugs will change at different times. Okay, so let's continue to read.
From what?
From however.
However.
From however.
Oh, okay. However, animal species, strain, gender, and age, as well as fertility and other biological cycles, represent additional sources of variability that must be controlled for. Results from experimental chronopharmacology studies have led to studies investigating the relevance of dosing time for drugs in humans. Drug chronopharmacology usually displays reciprocal 24 hours patterns in mice or rats and in humans, whose circadian physiology and clock gene expressions differ by nearly 12 hours with reference to the light-dark schedule. In rodents and humans, the daytime-dependent toxicity and/or efficacy of medications reflect circadian drug uptake and/or metabolism, circadian drug sensitivity of target cells and tissues, or both.
Okay, thank you very much. So I think this is probably clear. Any questions on this one? So one of the key points is that yes, there is a circadian influence on the administration of drug, on the toxicity of drug, but there are other factors that influence, that needs to be controlled for. And this is true for mice, but this is even more true for humans. because the considerable variation there is between human is more than the variations between mice. Usually when you study mice, you have mice from the same genetic background, they are in the same environment, they are in the same laboratory, they are given the same food, but when you have patients, they are all different. Okay? So, how could you do? I mean, you know that, first of all, you know that, for example, injecting a drug in the morning may be better than injecting a drug in the evening. Okay. But think about the patient. You have a patient that arrives, and it's a cancer patient, and you have to administer a drug. And you know that you may have a very high likelihood of having a complete remission if you inject at a certain time compared to a different one. And you need to make a decision. What are important? This is in the paper, but I want to think about, I want you to think about it. So you have a drug that has completely different outcome. It's efficient at the time and it's not efficient at different times. What are the things that you should know? Can you just say, okay, you are coming and I'm going to inject the drug in the morning because it's better? Is that enough?
You need to check the patient's sleep cycle.
You can check the patient's sleep cycle. Yeah. You can check the patient's sleep cycle, and it's important because maybe why it's important.
Depending on the cycle, that'll affect the rest of their rhythms, so it'll affect the enzymes in the liver as well.
Yeah. So actually what happens is that when you have a patient, you actually do not know what is the circadian time of your patient's body. Because maybe your patient is somebody doing shift work, and his biological clock is out of phase compared to a normal person. Okay? So what is important is not the time of day. What is important is the circadian time of your patient. Do you understand what it means, the circadian time? Okay, the body time of the patient. But checking sleep, it's not that easy to do. Okay, so of course you can follow your patient and see what happens. What else could you do? What information from your patient might be interesting to get?
Metabolites rhythm of the patient.
Metabolite?
The metabolite rhythm of the patient.
Yeah. So something that you could do, for example, what?
Measuring the concentration of metabolites in the patients.
Yeah, what metabolite?
For example, things that metabolites that is important for the drug, for the effect of the drug.
Yeah, that's a very good idea. So for some, there is a part of that on the paper. For some, especially for 5-fluorouracil, it is very well known. So since you come with it, let's go to see the picture here. This is on page 612. There is a big figure there. And you have a part that is dark blue, and this is the night, and you have a part that is light blue, and this is during the day. And at the outside of the circle, you have 5-FU, and you have at the lower part, at the lower quadrant, you have a different drug. And you can see the legend. Multiple coordinated mechanisms in the detoxification of anti-cancer drugs in healthy tissue. Selected example for 5-fluorouracil, an antimetabolite, and oxaliplatin, an alkylating agent, in nocturnally active rodent. Following rapid cellular entry, 5-FU is rhythmically catabolized by dihydropyrimidine dehydrogenase. The active metabolites that are rhythmically formed through pathways not shown here suppress DNA synthesis through inhibition of thymidylate synthase, an enzyme with rhythmic activity. 5-FU can also elicit apoptosis. Are you familiar with apoptosis? Yeah? A process that is antagonized by BCL2, an anti-apoptotic protein that is also rhythmic in bone marrow, a main toxicity target for 5-FU. Finally, proliferating cells that are more sensitive to 5-FU following exposure during the DNA synthesis stage as compared to other stages of the cell cycle. So you can see the 5-FU arrow that you have here, it's the best in the early hours of the day because DPD, dihydropyrimidine dehydrogenase, is high, TSS is low, and BCL2 is low. So this means that the outcome of 5-FU is the best at that time. Okay. So what you could do, as you suggested, for example, you can measure the amount of BCL2, you can measure the amount of DPD, and when DPD is high and BCL2 is high, this is the best time for 5-FU. You could do that. You could actually measure the enzymatic levels. But can you just measure one time? Can you get a patient, okay, give me your blood and give me tissue sample. First of all, tissue sample is very difficult, okay, but you could do. You need, is 1 point enough?
No. It should be a long time.
Yeah, so ideally, you should be able to know really what is the rhythm of these enzymes in your patients, okay? What they say, especially this thymidylate synthase, TS, it's an enzyme with rhythmic activity. And what you don't want to have is a high level of thymidylate synthase when you inject 5-FU. So what you should do really is to measure a complete circadian cycle of thymidylate synthase to know when is the best time. So you can see what is the problem. Is how do you know what is really for each patient the best time?
So do cancer cells have the same rhythm, and do they even have a rhythm as healthy cells?
That's an extremely good question. So for chemotherapy, there are two different things. First of all, there is the effect that you want, which is killing cancer cells, and the effect that you don't want is to kill healthy cells. Okay. So what is described here is only the toxicity. Okay. You want to inject 5-FU at a time where it will be less toxic for the organism. For example, you don't want to affect bone marrow, as they say. Because if 5-FU goes into the bone marrow, then you have a lot of bad side effects. Okay. What is very lucky, in a way, is that usually cancer cell metabolism will be completely different from the metabolism of the body. So even though the side effects are lessened, if you are lucky, this may mean that even though the anti-cancer effect of your drug will be the same at any time, but the toxicity of the drug will be lower at certain time. Okay? But the problem is that different cancers will also respond differently. So that's also one of the big problems. So currently what they are doing with these is that they are using different things, as you said, enzymatic activity and things like this. They also measure cortisol rhythms to determine what is really the best time for drug administration. And something that this guy, this Francis Levy, has designed is actually a kind of injector that injects drug only at certain time. So that you don't need to have a nurse coming at the right time. You have an injector that is programmed to inject a dose of any anti-cancer drug that you can use at the precise time that is needed. Okay, so this is the kind of things I wanted to say. But the big problem is what markers you can use to know the circadian time of your patient. This is the very important thing. And as you said, one time is not enough. You need to know precisely what is the circadian rhythm of your patient. But they are trying to find a way by measuring as little point as possible, but make sure that this gives an accurate measure of what is the circadian rhythm of your patient. So they are trying to improve these ones. And all these things are currently under clinical trials. So hopefully in the future there will be something much more efficient for cancer. Okay, so that's the kind of last few points I wanted to to talk about. You can take a look at page 615, which is comments on the future. Is what we have kind of already been discussing. Time differs between individuals, and the future, maybe it's a custom tailored medicine for every single patient with a different clock, with slightly different rhythms. There will be a personalized cancer treatment. And this is of course not true only for cancer, but for many other diseases, because all the drugs that we take will be metabolized by the liver. So maybe for every single drug there is a best time of administration. Okay. And we still have time, so I will read this paragraph just for the fun of it. Humans can be grouped into different chronotypes, ranging from lags to olds. Patients can also display distinct patterns in circadian physiology, possibly related to the impact of the disease on their circadian timing system components. Hence, the physiological significance of daytime differs between human individuals. It is still not clear how much genetics contributes to these different individual behaviors. In the simplest case, the different circadian phases can be explained simply by period length. Indeed, a modest difference in period length can cause a large difference in phase. Unfortunately, the determination of period length of circadian behavior and physiology in human individuals is costly and time-consuming, and thus cannot be done on a routine basis in the clinic. Like, you cannot take samples of the liver of your patients to know what is the TS activity and the circadian, so it's very difficult. However, the recording of period length can be accomplished relatively easily on cultured primary fibroblasts obtained from skin punch biopsies using lentiviral vectors and compassing a circadian luciferase reporter gene. Such experiments revealed unexpectedly high variability between human individuals, although it is not yet clear whether fibroblast gene expression cycles correlate with human circadian physiology. Similar experiments with mice carrying mutations in CROC gene suggest that, at least qualitatively, fibroblast rhythms correlate with locomotor activity rhythms. In isolated case, circadian periods could also be monitored in primary keratinocytes from plugged hairs using lentiviral vector technology. Plugging hairs is obviously less invasive than skin punch biopsies, and it is hoped that the keratinocyte procedure can be optimized to make it generally applicable. If these attempts succeed, it should become possible to record circadian gene expression cycles from hundreds to thousands of human individuals. So it's not written, but what they're also doing is not keratinocytes, but it simply is mouth swabs. They actually collect the mucosa, and they actually quantify gene expression, like PER2 gene expression, from samples of saliva or sample of mucosa of the mouth. So all these things to try to actually measure precisely what is the gene expression rhythm of the patient, and therefore to know what is the best circadian time at which to administer the drug. Okay, so that's the last thing today. So next week we have the final test. You know already how it's going to be, so there is no problem. Okay, there is no problem. But, there might be newer questions, different questions, but you know how it's going to be. All right. So if you have any question, go for it. Otherwise, that's the end of this kind of discussion.
Is larks and owls actual scientific terminology?
Yes, actually.
What's between?
There are many papers actually, and using lark and owls as saying, you know, this patient is a lark, this patient is an owl. It's kind of a little bit colloquial, but it's more and more used for different chronotypes. Instead of saying, I'm a morning chronotype or I'm an evening chronotype, then more and more lark and owls are being used. By the way, just for fun, please connect to the a big chronobiology kind of a questionnaire that is from Europe, but it's a worldwide thing. So just spend time to fill questionnaire. They are trying to get information on what is the different chronotypes in the world. And things. So it's very, very nice.
I think I've done it.
You have done it.
Yes.
So there may be something similar in Japan as well. But this is just trying. So anyway, thank you very much for coming to this lecture. Sorry, it's a little bit lack of structure, but oh well.
Is there anything in between larks and owls or just larks and owls?
I think in terms of biological significance, there may be something, maybe like chicken or
I'm a mid-afternoon chicken.
lark and owls and this nightingale and bats. Go on, it's a different type of animal.
That's true. Are there any marsupial birds?
Eh?
Because like bats are like Clyde Island.
Yeah, bats, they are dusk, right?
They're just in dark, I think, aren't they?
Are they? There is a name for this kind of animal, are there? Huh?
I think it's Crespula. Crespula, something like that.
Ah, crepuscular.
That's the one.
Crepuscular, yeah. Sounds like fire.
It's amazing how these different birds.
Yeah, it's crepuscular, it's the same name.